Mitosis and meiosis. Welcome to another video of How to Science 123. Today we're going to be going over mitosis and meiosis. Before we really dig into the nitty-gritty of those topics, I do want to go over why cells divide. Cell division is very important for a multitude of things, but I think the most important things include reproduction and renewal and repair. The idea of reproduction, for example, in us humans, we are multicellular eukaryotes, so we go through sexual reproduction. We need our cells to divide in order for embryonic development to occur, right? If we want to make a baby and a baby to actually exist, our cells, the cells are going to need to divide in order for that baby to actually look like a baby. Um, Another big important fact is renewal and repair. If we get a paper cut on our skin, our cells are going to need to divide in order to close that cut up. So there are a multitude of reasons as to why cells divide, but I feel like the two important ones are reproduction and renewal and repair. I also want to go over the idea of sister chromatids because we'll be seeing these a lot when we get into mitosis and meiosis. So sister chromatids, this is a pretty important graphic, I feel like. People kind of get this confused because they assume that this is one whole chromosome, right? It looks like an X. But this is not one whole chromosome. This is actually referred to as a sister chromatid because they are attached. So one chromosome would be just this one right here, and another chromosome would be just this one right here. But since they are attached, right, they are attached at the centromere here, which is this waste part, they are referred to as sister chromatids. So in preparation for cell division, DNA is replicated and the chromosomes condense, and each duplicated chromosome has two sister chromatids attached along their lengths by cohesins. This is referred to as the centromere. It's kind of like this narrow waist, referred to it kind of like as a belt of the duplicated chromosome, where the two chromatids are most closely attached. And when we get to cell division, these sister chromatids are going to separate from one another. So essentially, you see this line right here. When we get really into mitosis and meiosis, there will be specific parts in those developments that cause these two sister chromatids to basically pull away from one another. To explain why this is referred to as sister chromatids, remember we talked about how DNA replication is extremely important. When we get into concepts like the cell cycle, you know, DNA replication is always happening in our bodies, right? For epithelial cells or skin cells, they are constantly dividing. And when we go through DNA replication, we're basically replicating this entire chromosome, which is what contains our DNA, into another chromosome, right? And these are going to be attached to each other, which is why we refer to them as sister chromatids. If these two were not attached, right, if they were not, like this, then we refer to them as homologous chromosomes. But since this chromosome is the exact same as this chromosome, because this one was copied and, basically printed out from this chromosome, right? Imagine this is the template chromosome, and this chromosome was the essential copy of it. See how they are attached down this line? This is what is referred to as a sister chromatid. But if I were to separate these two from one another, which we'll talk about later, then they would both become homologous. They are not homologous chromosomes until they are separated into the respective cells. Um, I also want to talk a little bit about, just eukaryotic cell division, which is basically the representation of mitosis, which is the division of the genetic material in the nucleus. Cytokinesis is actually where the division occurs. And also some important points, gametes are produced by a variation of cell division called meiosis, which we'll talk about later. We're going to first talk about mitosis. And meiosis yields non-identical daughter cells that have half as many chromosomes as the parent cell. We'll talk about those reasonings behind that later. Let's talk about the cell cycle. So I feel like this is a pretty good graphic when referring to the cell cycle. The cell cycle consists of the mitotic M phase, which is mitosis and cytokinesis, and interphase. Interphase covers about 90% of the cell cycle, and it can be divided into three phases. We have the G1 phase, we have the S phase, and we have the G2 phase. And the cell grows through all three of those phases, but the chromosomes are only duplicated during the S phase. So for a majority of a cell's life, it's not constantly replicating. It's going to grow to a significant amount. It's going to replicate its DNA and then go into mitosis. Because remember, right, if we're going to divide a cell into two, it's going to need to have enough nutrients to support two cells. If we divided this cell right here at the G1 phase, it's not going to have enough nutrients to equally, you know, have nutrients for one part of the cell and another part of the cell. So interphase is very important before the mitotic M phase because this is where the cell grows, this is where the cell matures so that it's able to go into cell division and support two new cells. Okay? The G1 phase, if you see a G next to any of the phase, the biggest point of that phase is for growth. The S phase is the most different phase compared to the G1 and the G2 because this is specifically meant for DNA synthesis, DNA replication. We're trying to replicate the chromosomes so that each new cell, when we get to mitotic or when we get to mitosis, has the exact copy of DNA as the other cell. Let's talk about mitosis now. So mitosis is conventionally broken down into 6 phases. We have prophase, we have prometaphase, we have metaphase, anaphase, telophase, and cytokinesis. So I'm going to talk about each phase here and just sort of describe what actually happens in each of these phases. So we talked about the interphase, right, which is where a cell spends 90% of its life before it goes into mitosis. Well, let's say the cell is at the G2 phase now, right? It has grown to a significant amount, a sufficient amount, and it's basically saying I'm ready to divide. So now we're going to go into the mitotic M phase, and we have prophase, prometaphase, metaphase, anaphase, telophase, and cytokinesis. Let's talk about prophase first. Prophase is the first step of mitosis, and as you can see, we have the chromosomes, and they're sort of in the nuclear envelope. They're in the nucleus, and they're basically getting ready. And the prophase is very important because this is where we start. It's the initial part. It's the the initial thing of metaphase, right? We're seeing the chromosomes starting to condense. We're starting to see the nuclear envelope, which is holding these chromosomes, start to, break apart. And we're also seeing these structures referred to as centrosomes, which I'll have a dedicated slide to in explaining what these are. But we're seeing these centrosomes start to have these long finger-like things coming out, and these are referred to as microtubules, and these will be important in the next few steps. Prometaphase, in my opinion, is one of the more, it's kind of mixed in the science community on whether it should be included or whether it shouldn't be included. But the whole point of prometaphase is just to sort of continue off of what prophase was doing, which was getting rid of the nuclear envelope and condensing those chromosomes even more. So at this point, our chromosomes have fully condensed and we don't even see the nuclear envelope. It's very, just fragments of it at this point. We also see the centrosomes here at this point. They have migrated to opposite poles of the cell. And this is going to be very important when we get to metaphase. At metaphase at this point, is, I think, one of the most important steps. I mean, all steps of mitosis is the most important, but for me at least, metaphase was the most recognizable to me. It was the one that I felt like I knew at all times because it just felt the most recognizable. But metaphase is where we see a big variable change. We're going to see all the chromosomes lined up in the middle, and these long finger-like structures, which we were talking about earlier, which we refer to as microtubules, they're basically attaching to these chromosomes, and they're just lining them all up in the middle at this point. We refer to this as the metaphase plate. It's an imaginary plate, basically. Just imagine chromosomes lined up on a plate almost. Um, and these microtubules, which are coming out from the centrosome, they're basically attaching to each, for example, each sister chromatid and lining them all up along the middle of the cell. Anaphase is where we actually pull them apart. So remember, we had sister chromatids here, right? We talked about in the last slide. These were sister chromatids, and I said, we're going to separate these two so that they turn into homologous chromosomes. That's what's going to happen here. Anaphase, remember we had one centrosome on one side of the cell and another centrosome on another side of the cell. They're both going to depolymerize and basically pull each sister chromatid away from one another. So now we have an equal set of chromosomes on one side of the cell and equal set of chromosomes on the other side of the cell. And we're finally going to get to telophase and cytokinesis. Telophase is where we see the nuclear envelope starting to form up again. The chromosomes are starting to decondense, and we're seeing this sort of bump almost, or this sort of furrow or cleavage furrow actually, which is a very important sign of cytokinesis, right? We're seeing the cell sort of split itself into two, right? And we talked about how one half of the chromosomes went to the other side and the other half of the chromosomes went to the other side, right? Now we have an equal set of chromosomes in one cell and an equal set of chromosomes in the other cell. And now the cell is going to basically go through interphase again and repeat the process all over again. So this is mitosis complete. And this is sort of, I guess, a molecular image of it under a very, 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 very strong microscope. And you kind of just see the same thing here, right? Interphase, the cell is grown to a sufficient amount. Prophase, we see the centrosome starting to, work to, you know, go to the opposite poles of the cell. We see the microtubules starting to form. Prometaphase, same idea. The chromosomes have condensed at this point, and the centrosomes are moving to the opposite sides. Metaphase, the chromosomes are all lined up, and we have what's it called? We have our centrosomes on opposite sides of the cell. And then anaphase, we see them being pulled apart. And telophase, we see the cleavage furrow kind of thing right here. And we see the cell split into two. So this is what I wanted to get into. In the last slide, we talked about centrosomes. I'm going to go in depth into it here. This entire structure is referred to as the mitotic spindle. The mitotic spindle consists of what we refer to as microtubules, the aster, and the centrosome. So in animal cells, assembly of spindle microtubules begins in the centrosome, a type of microtubule organizing center. The centrosome replicates during interphase, forming two centrosomes that migrate to opposite ends of the cell during prophase and prometaphase. So if we look at this last slide here, right, we have two sets of centrosomes. We have one here, and then we have another. In the G1 phase, which is kind of where this cell is going to be after it goes through mitosis, we only have one centrosome. But when we get to DNA replication, we're going to replicate these chromosomes. So now we have sister chromatids, and we also replicate centrosomes. So now we have two centrosomes. And those centrosomes are going to be vital when we get to the next few steps of mitosis, right? In prometaphase, metaphase, we're going to need two centrosomes to pull the sister chromatids away to opposite ends of the cell. Okay? And these are some important things that I want to talk about here. Kinetochores specifically. So we were talking about the centromere region here, which was this sort of waist kind of region of the sister chromatid. There's actually a protein complex referred to as a kinetochore. And you might think, all right, these microtubules, right, they're attaching to the centromere, which is, a fair assumption because it is technically the centromere, but on top of the centromere is this protein sort of just protein type structure referred to as a kinetochore, and that is where the microtubules attach. The microtubules attach to something called the kinetochore, which is sort of resting on top of the centromere on each, specific on the sister chromatids. So all in all, the mitotic spindle, right? We have the two centrosomes, then we have the microtubules, which are these finger-like structures which we're talking about. The aster, which is just this glowing part, as you can see, it's just kind of like a radial array of short microtubules, which just extends towards the top, basically, right? This green, you see the glowing part? We refer to this as the aster. And then we have our kinetochore microtubules. These are microtubules that directly attach to the kinetochores. And then we have our overlapping non-kinetochore microtubules. So some microtubules, they just won't attach to any chromosomes because a lot of them come out of the centrosome. And these are referred to as non-kinetochore microtubules. They're just, extending as the centrosome, does its job. And then this is cytokinesis. We talked about the cleavage furrow here, which is just this sort of rigid, kind of like bump-like structure where it just seems like the cell is starting to split. I kind of like to imagine it as like it's just like a butt, basically. Like this is just kind of the butt area, kind of. I know that's kind of crazy, but it's just kind of how I imagine it, right? If you were to see the cleavage furrow of an animal cell, right, this is an indication of the cell splitting into two. And so, I mean, that's just one way of memorizing it or remembering it. There's multiple ways that you can think of it. I think of it like that. And the first sign of a cleavage is the appearance of a cleavage furrow, a shallow groove in the cell surface near the old metaphase plate, right? So this was the old metaphase plate where the chromosomes all lined up. And now the cell is going to split into two right where that old metaphase plate once was. And this is what is the final step of basically mitosis, is cytokinesis. We're seeing the cell split into two new daughter cells. And also, just an important fact here, we're talking about plant cells in this case here too, right? Plant cells are going to need to develop their cell wall. So we're going to see the cell plate start to form here as the daughter cells get replicated. And the cell plate is going to turn into two separate cell walls, and now we're going to see the cell split into two. So vesicles will form the cell plate. We have the wall of the parent cell. We see the cell plate formed. And now we have a new cell wall for one daughter cell and then the other daughter cell. I also want to go over the idea of the cell cycle control system. So the sequential events of the cell cycle are directed by a distinct cell cycle control system. The checkpoints are subjected to both internal and external regulation. So kind of think of it as like the control system of a washing machine. It proceeds on its own, driven by a built-in clock, and has specific checkpoints where the cell cycle stops until a go-ahead signal is received. One example of a checkpoint is the maturation promoting factor, which is basically this G2 checkpoint into the M phase. So we basically see this cell, right? It's going to get to the G2 checkpoint here, and it's going to basically, at this point here, the maturation promoting factor, which is just the concentration of cyclin, in order for the G2 cell to get into the M checkpoint, or get past the G2 checkpoint, the cell's going to need to know if it's grown to a certain standard, right? Like we said, we need to make sure the cell has enough nutrients to support two new cells. So this buildup of this cyclin concentration as the cell cycle progresses forms this complex known as the CDK cyclin complex or the cyclin CDK complex, which creates this maturation promoting factor to allow the cell to get past the G2 checkpoint. And then once we get into mitosis and we see the cell actually divide, the cyclin is degraded and it restarts the cycle all over again for the cell to go back into, um, mitosis. So in order for a cell to make it past the G2 checkpoint, it needs to pass the test of being mature. It needs to pass the test of being big enough and being able enough to go into mitosis, right? Because the most important thing about mitosis is we're basically creating two new cells, and if from one cell, and so if that one cell doesn't have enough nutrients to create two new cells, then it won't make it past the G2 checkpoint. It'll go through apoptosis, which is just a cell's programmed death. So the MPF, or the maturation promoting factor, which is just the buildup of cyclin concentration, is really important because it allows the cell to basically build up its processes and basically tell or signal that it's big enough or mature enough to go into mitosis and divide. Some other checkpoints as well here too. We have one at the G1 checkpoint, which we looked at earlier, which basically just tells the cell it basically signals that it's ready to go into DNA synthesis, right? Without a go-ahead signal, it enters G0, which is just a phase where the cell doesn't do anything. It doesn't go into cell division or anything like that. Some examples of G0 cells include our neuron cells, right? At some point, our neurons are going to stop, growing and dividing because that's just how our body biologically works. So they will always be in this G0 phase. They won't go past this point. And if it does go past the go-ahead signal, then it will continue the cell cycle to go into DNA synthesis and divide. And then we have the M checkpoint here. So For example, in prometaphase, the cell receives a stop signal when any chromosomes are not attached. So if none of these chromosomes are attached to a microtubule, then the checkpoint will basically say, all right, you can't go into cytokinesis because that means the cells are not going to separate to their opposite poles. But when all chromosomes are attached, then the cell will go into metaphase, and then it will go into anaphase to allow the chromosomes to enter their opposite side of the cell to go into cytokinesis. We talked about mitosis, now it's time for us to go into meiosis. So this I feel like is a pretty cool image when it comes to sort of talking about meiosis. It's just a very good summary, a very basic summary of what happens. So I'll briefly go over this image before we get into the nitty-gritty of meiosis. So meiosis basically is the creation of our gametes, right? We need gametes. Gametes, for example, are sperm cells in males and eggs in females. So, for example, me as a guy, I will go through meiosis and I will basically have one homologous chromosome from my father and one homologous chromosome from my mother. And these chromosomes are going to duplicate, like we talked about with the S phase and DNA synthesis. They're going to duplicate, so now I have a sister chromatid and another sister chromatid. We're going to go through meiosis 1, so we're going to split the cell into two, so the homologs are separated, right? These two are both homologous chromosomes, but this is a sister chromatid and this is a sister chromatid. So we're basically going to separate those homologous chromosomes from one another. So now we have a sister chromatid in this cell, we have a sister chromatid in this cell. And now we're going to split the cell again. So now we have one homologous chromosome here, another homologous chromosome, another homologous chromosome, another homologous chromosome. So now we have 4 chromosomes to create sperm cells. These are all sperm cells. And then meiosis, for example, a female, it'll create eggs, and then these sperm and these egg cells, if sexual reproduction does happen, they will combine with one another to create a fertilized egg, which is referred to as a zygote. So meiosis is basically mitosis twice. We have one where we separate the homologous chromosomes, and then another where we separate the sister chromatids. Let's get into it. So I do want to talk about a little bit about, somatic cells and gametes, so which we'll get into right now. So somatic cells are the cells that are basically not sex cells or sex-related cells. Gametes are reproductive cells, like our sperm cells and for males and the eggs for females. Anything that isn't, related to sexual reproduction or sexual reproductive cells are referred to as somatic cells. So the sex chromosomes, which determine the sex of the individual, are called X and Y. So human females have a homologous pair of X chromosomes. They have 2 X chromosomes. And then human males have 1 X and 1 Y chromosome. And the remaining 22 pairs of chromosomes are called autosomes. So in total, we have 23 pairs of chromosomes, but only 22 are referred to as autosomes because they don't relate to the sex, basically, or the sex of the individual. And each pair of homologous chromosomes includes 1 chromosome from each parent. So we have 1 from mom and we have 1 from dad. And the 46 chromosomes in a human somatic cell are 2 sets of 23, right? We have 1 from mom and we have 1 from dad, which basically gives us 46. A diploid cell has 2N, 2 sets of chromosomes, and for humans, the diploid number is 46. So 2N is equal to 46. So that means N is equal to 23, right? We have 23 from mom, and then we have 23 from dad. So let's go in depth into what a gamete is and the idea of haploid cells. So a gamete, sperm or egg, contains a single set of chromosomes and is thus a haploid, right? We refer to as a diploid as 2N. But in this case, we're referring to a haploid, which is just N, right? Because haploid sperm or egg cells, right? Sperm cells, they're going to only have DNA from the father. The egg cells are only going to have DNA from the mother. But when they combine, right? Because a sperm has 23 and then an egg has 23, when they combine, they create 46, which will give us the diploid cell. So for humans, the haploid number is 23, and each set of 23 consists of 22 autosomes and a single sex chromosome, right? We have 22 autosomes and one sex chromosome. So if we're a male, that could be an X and a Y when those two combine, but for females, it'll just be an X and an X. In an unfertilized egg ovum, the sex chromosome is Y, and a sperm cell, the sex chromosome may be either X or Y. So for sperm cells, we'll have 22 autosomes and one sex chromosome, so that could be either X or Y. And let's say the egg had an X chromosome, so if the sperm cell had an X chromosome and the egg had an X chromosome and those two combined, the sex cell, right, two chromosomes adding up together, X and X, will give us a female. And this is a pretty good diagram here. We have the centromeres. So we have three maternal sets of chromosomes. We have 1, 2, 3. And then we have three paternal sets of chromosomes, 1, 2, 3. So 2n is equal to 6. So we have 3. If we're to split these into 2, we'd have 3, right? We'd have 3 from the mom and then 3 from the dad. But then the total is equal to 6. And these are homologous chromosomes. These are sister chromatids for one duplicated chromosome, but we have homologous and homologous. Fertilization is a utilization of gametes, the sperm and the egg. So the fertilized egg is called a zygote and has one set of chromosomes from each parent, right? The sperm, it contains one chromosomes, and then the the egg contains the other chromosomes. You add them together, you get 46. 23 plus 23 is equal to 46. And the zygote produces somatic cells by mitosis and develops into an adult. So basically, we're going to go through meiosis, right? The meiosis in the testes for the male to create sperm cells. We're going to go through meiosis in the ovaries for the female to create the egg. And these both have 23 chromosomes, right? We have 23 chromosomes from the dad, and then 23 chromosomes from the female, from the mother. And then they're going to go through fertilization because they're going to combine together. The sperm is going to enter the egg, and that's going to create what we refer to as a diploid zygote, right? Because we had 23 chromosomes from the father, 23 chromosomes from the mother. 23 plus 23 is equal to 46, so 2n is equal to 46. And this will go through mitosis and development, so this cell will just keep dividing and dividing and dividing and dividing until a baby is formed. So remember in the past slide, I showed you meiosis 1 and meiosis 2. This is where we get into those nitty-gritty topics. So division of meiosis 1 occurs in 4 phases: prophase 1, metaphase 1, anaphase 1, telophase 1, and cytokinesis. Prophase 1 is actually where a lot of important things happen. One of the biggest important things is referred to as crossing over, and we'll get into this. So let's talk about crossing over. So after interphase, the sister chromatids are held together by proteins called cohesins. So these are sister chromatids, and they have these cohesins, which are just holding them together. The non-sister chromatids are broken at precisely matching points. So these two are non-sister chromatids, right? This is the maternal sister chromatid, and this is the paternal sister chromatid, but they're non-sister chromatids, right? This DNA break, it's going to break this part of the paternal sister chromatid, and it'll break it at the exact same spot in the maternal sister chromatid. And basically, what's going to happen is this thing called the synaptonemal complex is going to start forming, and that's what's going to hold these homologs tightly together. So the maternal and the paternal, they are referred to as homologous chromosomes. And basically, what's going to happen is the DNA breaks are going to be repaired or during synapsis, but this is where crossing over happens, right? We talked about the breaks here. What's going to happen is this DNA from one non-sister chromatid is going to join with the DNA from the other non-sister chromatid. So the sister chromatid from the maternal set is going to combine DNA with the sister chromatid from the paternal set, and it's going to create these structures called chiasmata. Chiasmata. And that basically produces recombinant chromosomes, which combine DNA inherited from each parent, right? So we have DNA inherited from the mom and the dad. It's going to create some genetic variation, which is Very important. Genetic variation is important. And that's where crossing over happens. Crossing over happens in prophase one. We talked about the chiasmata forming here. We talked about the cohesins. We talked about the DNA breaks and then the synaptonemal complex, which is forming and just basically joining these two non-sister chromatids together to combine DNA. So X-shaped regions called chiasmata are sites of crossovers, right? This is an X-shaped region and this is an X-shaped region. These X-shaped regions are chiasmata. That's where crossing over has occurred. And in metaphase 1, these pairs of homologous chromosomes line up at the metaphase plate with one chromosome facing each pole, right? So the chiasmata, these two combined things are still there, but the homologous chromosomes are going to be lined up right in metaphase 1. We talked about this in mitosis. These centrosomes and their microtubules are going to line up these chromosomes in the middle at this metaphase plate, this imaginary metaphase plate. But in metaphase 1, right, it's different in mitosis, right? We're just going to separate the sister chromatids. In this case, we're going to be separating homologous chromosomes, right? Because we have one from mom and we have one from dad. And so what's going to happen here is we're going to get to anaphase 1 and telophase 1 cytokinesis. Anaphase 1 is going to pull the homologous chromosomes away from one another. But look what's interesting here, right? Remember what we talked about with chiasmata and um, the idea of crossing over, right? The recombinant DNA, right? Between these two chromosomes interacting with one another through crossing over, we have some genetic variation, right? This homologous chromosome and this homologous chromosome, they're different. This chromosome contains some DNA from the maternal set, and this chromosome contains DNA from the paternal set. So this contributes to genetic variation. So now these homologous chromosomes, they're going to separate from one another. So now the homologous chromosomes are on this side, and then we have another set of homologous chromosomes on the other side, and we're going to go through telophase 1 and cytokinesis. The cell's going to separate into two new cells. Now we're going to do it again, right? Because we need to separate the sister chromatids from one another. So we're going to do the exact same thing, and instead of creating two cells, right, we're going to create four because we separated this one cell into two, and now we're going to separate those two cells again. So we're going to have 1, 2, 3, 4. And this will give us our haploid daughter cells. So in prophase 1, same idea. Centrosomes, nuclear envelope is being gone. The chromosomes are starting to condense. And we see it happening in both cells, right? After telophase, after cytokinesis and meiosis 1, we had two new daughter cells. And so in metaphase 2, we're seeing the same thing happen in the two new daughter cells. The centrosomes are going to line up the sister chromatids at the metaphase plate in both cells. And then in anaphase 1 and telophase, in anaphase 2 and telophase 2 in cytokinesis, we're going to see those sister chromatids separate from one another. We're going to see the centrosomes pull away the sister chromatids, and they're going to depolymerize, and they're going to go to opposite sides of the cell again. And so now this one cell is going to turn into two, and this other cell that we had after meiosis 1 is going to turn into 2. 2 plus 2 is 4. We have 4 haploid daughter cells forming. And if this was in a man, this would be sperm cells. This is in a female, this would be eggs. This is kind of just mitosis and meiosis sort of simplified, sort of comparing the two. So we talked about in mitosis, we have the parent cell, and then we have chromosome duplication. Biggest thing, the individual chromosomes are going to line up, but we're not going to see anything referred to as crossing over. We're not going to see chiasma form, and none of that happen. Like, that's not going to happen, basically. So basically, meiosis reduces the number of chromosome sets from 2 to 1, producing 4 cells that differ genetically from each other and from the parent cell. Unique steps of meiosis, if you really want to compare the two. In meiosis, we obviously have synapsis and crossing over in prophase 1, the alignment of homologous pairs at the metaphase plate, right? In mitosis, we're only lining up the sister chromatids. We're not lining up homologous chromosomes like we see here in metaphase 1, and separation of homologs during anaphase 1, right? In mitosis, we're going straight through it. We are not lining up any homologous chromosomes, and we're not separating homologous chromosomes. We're only separating the sister chromatids. And this is just some more help in understanding prophase 1 of meiosis. So we could see the pair of homologs, the non-sister chromatids, held together during synapsis. We see the chiasma form, which is just the X-shaped region where crossing over occurred. And then we see the movement to the metaphase plate. They're going to separate from one another, and they're going to separate the sister chromatids. And now we have 4 daughter cells, and they have recombinant chromosomes, some genetic variation. And that is mitosis and meiosis. Thank you for watching.